Okay, let's get started. All ready to go. All of our clickers out and ready. What's happening next Friday? Second midterm. It's the eclipse. I'm not sure the eclipse of what, but we'll talk about that again later. So again, same format, 50 questions, multiple choice, one single best answer you can have as long as you need. Anybody who's at DRC taking the test there, please send me an email beforehand to remind me to make sure the test gets over there. What's it going to be on? Second half of Chapter 5 and Chapter 6. So it's a less overall material, but because of that, it's going to be a little bit more in depth. Again, all of my questions come from lectures. They don't come from the book. So when I write the exams, I go through my lectures. I don't crack open the book and say, how can I find a really obnoxious, obscure question in here that I didn't talk about so I can make sure that people do really badly on the exam. That's not how I do this. Yeah? So the question is, I'll paraphrase again. Do I just go over the slides that I have, or I'm actually just going to cover the things which I talk about? I'm just going to cover the things I talk about. So if there are things that we don't get to, particularly RNA world stuff, potentially next Monday, that if we don't get through all of that, it's not going to be on the exam. And the latest new material will be on Monday of next week. So hopefully by then we'll have gotten through translation. I think we should have by then. Okay. And then, clicker questions are a good indication of kinds of questions I'm likely to ask on the exam. So let's have them. The binding of which of the following, oh, sorry about that. I'm sure this funky thing here. The binding of which of the following snRNPs to the transcript is probably the most important in terms of which exons will be used in the mature messenger RNA. U1, 2, 3, 4, or 6. Not 5. Ten, 5, 'Good. Looks like everybody's on the same page with this. Oh, here we are. Yes, it's U2. It's really about where you get U2 binding. That's going to bind to where in your intron? The branch point, exactly. So it's going to be binding to the branch point. It's binding to that branch point, which is then going to be the most important in determining which introns are cut out, and which introns are cut out is going to depend on which exons end up getting used. And so that's the key to that. What about U3? What does it do? It's. What about U3? It's not in the spliceosome. That is correct. What is it doing? It's involved in ribosomal RNA. So it's the cleavage part of ribosomal RNA that we talked about right at the end last time. So, and U1 is binding where? It's at the end of the exon or the beginning of the intron, depending on how you look at it. And U4, 5, and 6 are important for the actual enzymatic activity of cutting and rejoining, particularly probably U6, although that's still a little bit controversial. in terms of how that counts. Okay, so that's splicing. We'll now move to the other end of your messenger RNA, and that is that transcription termination in eukaryotes is due to RNA helicase, exonuclease, endonuclease, RNA secondary structure, or B and C activity. Yeah. It says termination. So we have a lot more differences of opinion here. So RNA secondary structure is important in what kinds of organisms? Bacteria. It's important in bacteria, not important in eukaryotes. So nobody likes RNA helicase, even though it's critical for what? RNA helicase is important for splicing. We have to have RNA helicases for splicing. So that leaves us with B, C, and E. So most people think exonuclease. Why? Okay, it comes in from one end. How is that end generated? So where's the end that exonuclease chews on? Where does that come from? What kind of activity do you need to get an end? You need an endonuclease activity to cut and give you an end to chew in from. If you don't have an end, the other end is the cap, which is a 5' end. So you need both. And when you look at old exams, you will probably see a question extremely similar to this, because I think it's something that I've asked a number of times in the past. So the answer is E. You need endonuclease and exonuclease activity. Okay, give people a chance to get up and run away. Yes? So someday right after this, I should do another clicker question, like 10 minutes into class. Yeah, no, that actually is a good point, and that's having to do with clicker questions and how to deal with clicker questions, etc. When we get to the end of the term, you'll all get a chance to do an evaluation, and I'm going to try and have my own evaluation as well, in addition to the normal one that you do online. And there, please give me your comments about clickers. Do you think it'd be best to have them all the way through, at the beginning, at the end, etc.? And there are arguments for both. So just please let me know what you guys think about that. And it may make a difference in terms of virology next term, for those of you who are thinking about taking it. So yeah, today we're going to start with this last part of the central dogma, going from RNA to protein. So it's all about that last step, translation. We're going to talk about the genetic code, ORFs. Who knows what ORF stands for? Yeah, so number of different ideas here. Good, that's why we're covering it. This is the open reading frame. It's one of the few exceptions to F being factor. So it's the open reading frame. And that determines, based on your open reading frame and the genetic code, what protein you end up getting out. We'll look at transfer RNAs, we'll look at transfer RNA synthetases, and then a little bit of the ribosome, and may or may not get to translational initiation today. We'll see how that goes. So the translation cycle, this should look really, really familiar because we've already talked about similar kinds of cycles for what? Transcription has initiation, elongation, and termination. What else has initiation, replication? Exactly. So we've got 3 different mechanisms, again, that whole central dogma, all of which have 3 separate areas. And so if you're thinking about trying to assemble all of this crazy information, alphabet soup, etc., splitting up into those 3 different groups might be a good way to thinking about it. So initiation, again, we'll talk much more about towards the end today or on Friday. This is all about getting the appropriate tRNA, messenger RNA, and basically putting the whole ribosome together. So this is really analogous to getting that RNA polymerase to the right place on the DNA for transcription. Really analogous to the origin, where you're getting all of your origin recognition complexes, helicases, etc. Elongation, cranking along your messenger RNA in this case. The messenger RNA is the template, so again, very analogous to transcription and replication. Some point you need to end, and then that's termination, and we'll talk about termination in more details. This is, again, very similar to what happens in transcription, a little bit less so in terms of replication because replication termination is a lot less well defined, particularly in eukaryotic systems. So how do you go from nucleic acid language to protein language? We've got 4 nucleotides and 20 plus, actually, amino acids. Yeah, mathematically speaking, it's really easy. You just have 4 to the n number of possibilities in 4 nucleotides. So if your genetic code was just 1 nucleotide, you could only encode 4 different amino acids. If you had 2, you could do 4 squared, which is 16, but that's still not 20 amino acids. If you had 4, it's 4 to the 4th. I need my calculator to figure that out. Far too many. 256, if I remember correctly. So 3 is what you need, but it's far too many. You actually have 64 possibilities with nucleotides to give yourself 20 amino acids. So that's, again, really seems rather inefficient, but it gets back to biology. It doesn't necessarily have to be efficient, it just has to work, because that's what's been selected for. Yeah?
A couple of your old exams say 61 codons.
Somebody been reading my old exams? How could they? So some of the old exams say 61 codons. Those are codons which encode for amino acids, which we'll get back to in just a second, because clearly it's not just the individual amino acids that you need. You also need some other things, and one obvious one is stops, and we'll get to that in just a second. That's the 3 normal stop codons. That's why we're down to 61. There's also a start, which you need, and so these are 2 particular very special codes that you have to have above and beyond the saying, okay, we've got these 20 different amino acids which are there. Another question which came up actually very early on when people were thinking about the genetic code was whether it was an overlapping code or a non-overlapping code. And that's basically shown down here at the bottom. Unfortunately, for some reason, they deleted this figure from the current version of the textbook, so I took this older one. Again, it's but here, if you have a sequence, this is just a sequence of nucleotides, 5' to 3'. This is 7 nucleotides. If you had an overlapping code, which again, 3 nucleotides per amino acid, here you have AGC, the first 3, then GCA, then CAT, then ATC, and TCG. This is a way more efficient way of getting your information out of that particular nucleotide sequence. And so it would sort of make sense, at least in theory, that this would be the way that you use it. Turns out, of course, that's not the way that this happens. We have an overlapping code, and it probably has to do with the, I'd say, non-overlapping code, which gives you way fewer amino acids from these 7 nucleotides at the beginning. But probably the major reason for this is that now if you have one change, for instance here, you have change of one nucleotide, and we've got this A, so it's a C to A transversion here. You've now changed 3 codons, or potentially 3 different amino acids, as opposed to just one, which you have in a non-overlapping code. And we'll take a look at that in just a second. It turns out that even those single changes are probably such where you don't have much of a change in terms of the chemical nature of your amino acid side chain, with just those individual ones. So probably the overlapping versus non-overlapping code has to do with having good fidelity, and then if you have some kind of mutation, have it not be that detrimental when it takes place. So this is the genetic code as we have in our textbook. Basically here, there are 4 red circles, and so these are probably the ones that Stedman thinks are important. But not so much the upper red circle, that arginine here. This one's only supposed to indicate that there are a few amino acids that have a whole bunch of codons for them. And so you have up to 6 codons which are going to code for one amino acid. More importantly are these guys down here. So methionine, just one. AUG, also the start codon. It's not always the start codon. We'll get back to that in just a second. And tryptophan, which also just has one codon, which is being used to make that. And then getting back to the question here right at the beginning, what about those extra 3, the non-protein coding ones? These are the 3 stop codons, again, in most of organisms that are using the standard genetic code, which is this, and standard just means it's what's present in most organisms. And curiously enough, the more research we do, the more we start looking at other organisms, we find some that are using different versions of the code. But they're all really minor variations of the code. I don't like this form of the code very much at all. I much prefer this form, which again is not in the textbook, but I think this is a lot better way to understand the whole idea of the genetic code and how individual changes in nucleotides don't make that much difference in terms of your actual protein, an amino acid, and the chemical nature of that amino acid. And so this particular genetic code is color-coded based on the chemical nature of the side chain. So the way you read this, and we'll talk about it in just a second, is the first letter is going to be here on your y-axis, the major axis, second letter here on the x-axis, and then the third letter here on the lower y-axis. So start out with something which is A, followed by U, followed by G, gives us methionine. But again, importantly here is if you look at all of the other amino acids here, turns out that all of the amino acids that have U in the second position are the same color. What's the color mean here? It means they're hydrophobic amino acids. So basically, you can change pretty much anything else and get a hydrophobic amino acid at that particular position. So it's a really pretty amazing system. And again, if you think about the evolution thereof, you probably started with a lot fewer amino acids and fewer codons, and that's then expanded to get us to where we are now. And Dr. Lehman in the chemistry department can tell you all kinds of really cool theories about how the genetic code came to be. It's well beyond the scope of this course, but it's a really interesting one. Again, just another example here. If we look at our friends, aspartic acid and glutamic acid, or arginines and lysines, they're all really close together in this version of the genetic code, which says, okay, a couple of changes are really not going to make that much difference in terms of the overall chemistry of any given amino acid side chain. Not surprisingly here, you'll notice these guys are all in blocks of 4. So block of 4 here, block of 4 here, block of 4 here. And what that says is the first two nucleotides of your codon are the most important. It's these big blocks and not the secondary ones over on the other side. And so this is basically to remind me that the amino acid language is different than the nucleotide language, so it's really a translation. Now, you've gone from nucleotides, which have potential base pairing interactions, to now amino acids, which are much more involved in van der Waals interactions, charge interactions, and hydrogen bond interactions. So multiple different interactions that you're having there. Very, very different. How do you have this wonderful translation take place? It's all, as I mentioned last time, the magic decoder rings, which are the tRNAs right here. How do we figure out what the genetic code was? So nucleotides to amino acids. This was a real major tour de force by a number of researchers, particularly Marshall Nierenberg then at the National Institutes of Health. Very soon after Watson and Crick came up with their double helical model for what DNA looked like, he was able to synthesize RNAs, and particularly RNAs that were all of one nucleotide. And the first one was poly-U, and it turns out you put poly-U together with purified ribosomes and all the other goodies that you needed, all of the transfer RNAs, and you got just polyphenylalanine. And you can do the same thing with A's, you can do the same thing with G's, you can do the same thing with C's, and you immediately have 4 of the members, of the genetic code. The other ones are considerably harder, but eventually figured out basically just by synthesizing all of these nucleotides, you could put it all together. So because of this, UUU is phenylalanine. Also the single letter code is F. And what's the adapter? So you know UUU is phenylalanine. It's the tRNA. So before we talk about tRNAs, I wanted to get to these Fs, which are not factors, so the open reading frames. And because you have a non-overlapping code, it's really critical where you start. Because if you start at a different place, you end up with a totally different code. And so this is just an example right here of 3 different open reading frames that you have on one RNA. If you think about DNA, you've of course got 2 strands. So you've got 3 open reading frames that are potentially going in one direction and 3 going in the opposite direction. So any particular piece of DNA can actually encode 6 different amino acid sequences. So again, just depends on where you start. Up the top here is what's the classic open reading frame. And how do you open a reading frame? You open a reading frame by having a start codon. So here, methionine, followed by a certain number of amino acids. And again, it's kind of up in the air. People will make usually pretty arbitrary decisions on how many amino acids you have to have between a start and a stop before you call it an open reading frame. Most of the time, that's going to be in the order of about 50 amino acids. So if you have less than 50 amino acids between a start and a stop, most people are not going to consider that an open reading frame, although we're finding that there are more and more short peptides that actually are functional, active, etc. So that number is actually getting a little bit smaller here. If you have a different open reading frame, just here, for instance, you start at nucleotide 2. Now you've got a bunch of stop codons, all over the place here. No start codons to speak of. So here, there would not be a clear open reading frame, at least not in this particular sequence. Down at the bottom, however, if you start at nucleotide 3, and 3, 4, 5, 6, 7, 8, etc., there's no stop codon here, but there's also no start codon here. So this could be part of an open reading frame, but you wouldn't call it an open reading frame because you don't have a start. Starts, again, anytime you see quotes in one of Stedman's slides, it means be wary, there be dragons, or something similar to that. Start can be multiple different codons in bacteria. Usually it's AUG, but there are a number of cases where we know about protein sequences which have started from a GUG or a UUG start codon, Curiously enough, these guys, even if you use GUG or UUG, always has methionine that binds to that. So even if that's a particular different start codon, you always have methionine that starts there. Always AUG in our eukaryotes. Again, it's in quotes, 99.9 something% of the time. But there are some examples where it's actually not used. Stop codons, UGA, UAG, and UAA. Again, true in most organisms most of the time. But again, you've got variations on that. The last thing down here is only one reading frame contains a message that's actually used. This is fine for most cellular organisms. Turns out there are lots of viruses that don't bother with this at all. They say, heck, we're going to use multiple different open reading frames. And one of my favorites actually uses all three of the open reading frames to code protein that it does still need for function. So beware about that only one reading frame containing message. So what's our decoder ring look like? Hopefully most of you have seen the nice clover leaf structures, which is not at all what tRNAs actually look like. We'll see on the next slide what they really look like. But this is a nice way of looking at them. You've got from a 5' end to a 3' end. These are totally normal nucleic acids, 5' to 3'. In eukaryotes, they're made by RNA polymerase III. Have a specific sequence, lots of base pairing interactions, not always your standard base pairs. Here you've got GCs, but here's a GU. And there's always this CCA sequence at the 3' end of your tRNA. This 3' end, like everything else, has an OH on it. That OH then gets attached to the amino acid when you have this as what's called a charged tRNA. Another thing I wanted to mention with these RNAs is they've got lots of modified bases. And we'll look at some of those modified bases a little bit more further on. Dihydrouridine, methyl G, dimethyl G, pseudouridine, etc. So lots and lots of modified bases that you have in these tRNAs. So not unlike what happens in the ribosomal RNA that we talked about last time. There are very specific mechanisms for making these changes, sometimes through a guide RNA, like we talked about last time, the SNO RNAs, sometimes just specific protein-only enzymes that will be making these particular changes. You always have these base pairing interactions here with a D-arm, a T-arm, and this anticodon loop. One of the important things to think about with this anticodon loop is it's going from 5' to 3'. And so if you look here, there's a inosine, which we'll talk much more about later, at the first position of your anticodon. And in this case, where you've got an alanine tRNA, it's inosine, guanine, cytosine, positions 1, 2, and 3. So again, I promised I'd show you what this really looks like. Here's the first structure for any tRNA. This particular one is the phenylalanine tRNA. The structure was determined by Sung-Ho Kim at Berkeley a little before I actually got there, not that much long before I got there. But here, the important thing is that these are base paired, but they're clearly helical. You see all of these helices here. Base pair, helix, helix, helix. But it's not 4 separate helices. It's really just 2. There's this one helix here, which has the anticodon loop down here at the bottom, and another helix here at right angles, which has your amino acid hooked up to the end of it at the 3' end. So this is what the real structure of a tRNA looks like. And the D-arm and T-arm here. So D, by the way, I forgot to mention before, but this is where you always have a dihydrouracil. That's why it's called the D-arm. And T-arm is actually usually the T-psi-C arm, because there's always a pseudouridine, which is present in this arm. These guys wrap up together. So if you look at how this L structure comes from the cloverleaf structure, it's this helix which goes over into this helix. So green goes over into gray, and then blue lines up with your yellow at the end here. So that's how they're folding up together into this three-dimensional structure. Already mentioned 61 codons. He said 64, and now he's saying 61. What's the deal with all this stuff? Again, it's 61 codons which in the normal genetic code are encoding amino acids. In theory, if you've got 61 codons, you're going to have 61 tRNAs, right? So, these three nucleotides, each time you have anticodon-codon interactions, you're going to have 61 of them. Well, that's actually not the case. In fact, I don't think we've found any organism that actually has 61 different tRNAs in it. It's much more about the individual amino acids. So, in theory, you could actually just have 20 tRNAs, but then they would have to be recognizing these codons and anticodons in a very different way. Turned out it's between those two. It's between 20 and 61, usually between 30 and 50 of these. And the way that this can work is because of the Wobble hypothesis, which Francis Crick came up with not that long after he and Watson had come up with the model for the structure of DNA. And basically what this Wobble hypothesis says is that the first position of the anticodon, which is base pairing with the third position of your codon, because again these are always anti-parallel to each other, that doesn't always have to be a normal base pairing interaction. And this was the prediction that was made way before we had any kinds of structures for any of these things. And it turns out when we got the crystal structure of the ribosome, which about 15 years ago now, people found that this was exactly right. So that the interactions between the first position in your anticodon and the last position in the codon were pretty flexible, which, again, you could kind of guess from just going back and looking at the genetic code and say, hey, okay, well, all these things are coming in blocks of 4. That means the last nucleotide in the codon is not as important. Now, there are a couple of important things here. Again, we don't have our normal base pairing interactions all the time. Here, UA, this is exactly your normal base pair. You have UG, which can form. But most importantly, and as I pointed out on that previous slide, particularly for the alanine tRNA, very often at the first position of your anticodon, you have inosine. And inosine, as we'll see in just a second, can base pair with basically everything except inosine. So certainly you can find really nice IU base pairs, IC base pairs, IA base pairs. Occasionally you'll see IG base pairs, but that's really quite rare. So how do you get tRNAs? Well, we mentioned already you've got guide RNAs, which are important for getting these modifications. RNA polymerase III in eukaryotes makes them. In bacteria, it's just the normal RNA polymerase itself, which is making these tRNAs. All these tRNAs are made, just like ribosomal RNAs, as longer primary transcripts. They then get clipped at the ends and modified. That clipping, curiously enough, turns out to be done by another ribonucleoprotein complex called RNase P that was one of the other first examples for self-splicing, or RNAs which had enzymatic activity, because it cuts off the end, and it's clearly RNA which is doing that job. There also are many RNAs that have extra stretches of nucleotides, particularly in the middle of them, and that's what's shown here in this image. We have the normal RNA in gray, and then this extra piece here down at the bottom in blue. So many of these RNAs have extra pieces in the middle of them. Now, quite why that is not entirely clear, but it probably, again, has to do with the evolution of the genetic code. And it turns out in some organisms, you can actually have two separate pieces of RNA that then have to come together to form one functional tRNA. And that's probably what the original case was. You had two separate pieces that then came together. Those then got linked to each other. And in some cases, there's an intron which needs to get cut out. And so these are introns in tRNAs, not just introns that you have in messenger RNAs. So you have introns in messenger RNAs and in tRNAs. There aren't any examples that I know of introns in ribosomal RNAs, however. Yeah?
So those, you're saying that the introns in tRNAs are spliced out before the tRNA can function, or can they function despite the intron?
Okay, so the question is, can the tRNA function with the intron in it? And the answer is no, they cannot function with this intron in it. And to some extent, this actually seems to be a quality control, making sure you have all the modifications that you have to have in the tRNA, because the tRNAs are so absolutely critical for translation. If you have one which is wrong, then you're going to mess up all the proteins that would be using that particular tRNA. And so this process of having splicing, how you have to take out different pieces, have to modify all the rest of your tRNA, is probably very important for making sure that you have the right tRNAs at the end. Yeah?
Is there a risk of it having such a changed structure because of an extra intron that it has a different function than it was?
So the question is, with an intron, could it actually then start out having a different structure, which would again give you different structure, different function? And the answer to that is, as we'll see, when you look at the ribosome, the structure of those tRNAs has to be very, very consistent in order for it to work at all in translation. So you had another question, or is it basically the same one? Okay, and then I also need to put in a couple of plugs here. Again, Dr. Lehman here in the chemistry department is very interested in how this splicing event is taking place. And Dr. Wada-Royal in the chemistry department is very interested in how some of the modifications take place to some of the individual nucleotides that are present there. And in fact, he and I are now working on a particular project of a very strangely modified tRNA that you only find in Archaea. And quite why you do that is not clear at all. It may have to do with why Archaea are ancient. But again, different story completely. Turns out that a number of these tRNA modifications are absolutely critical. If you have a defect in, usually again, the enzymes which are involved in making some of these tRNAs, you have all kinds of problems. Again, some of these are really critical for health. And it turns out that a number of these are also why you have to have certain vitamins, because it turns out a lot of the vitamins which will then feed into some of the modifications that are happening in some of these transfer RNAs. We know more than 50 different modifications in just human tRNAs. Turns out that if you think about all different tRNAs, there are about 150 different modifications that happen to them. So really some of the most heavily modified RNAs that are out there. Again, some of the examples for that, dihydrouridine, also D, which is always present in that D-loop structure. Basically, all you've done is added a couple of hydrogens across the double bond, which is normally here. Dimethylguanine, just dimethylguanine. You also have some cases where you have sulfur, which has replaced the oxygen, thiouridine, and then inosine, our friend here, which is really important at the first position of the anticodon. Inosine, in terms of its structure, looks like deaminated adenosine. So if you look at the adenosine structure, pardon me, or is it down here at the bottom? Adenosine, in the absence of this amine group, gives you inosine. And inosine can base pair nicely in a semi-Watson-Crick fashion. It's actually really close to your normal base pairing interaction, slightly off in terms of where your riboses are, but really very similar. Look here with the C1 position, C1 position, C1 position, C1 position. That's your ribose, C1 here. C1 position, C1 position. It can base pair to pretty much everybody in a very, very similar kind of format. And so that's why, in many cases, we have inosine here at the 5' position in your anticodon loop. So that's RNA interactions between messenger RNA and tRNA. How does the tRNA know what amino acid needs to get put onto this? So these are our aminoacyl tRNA synthetases, which, again, chemistry department here, John Perona and his lab work a lot with these guys. So the amino-acyl tRNA synthetases, unlike the 30 to 50 tRNAs that you have to cover all 61 amino acid coding codons, here it's pretty much one amino-acyl tRNA synthetase per amino acid. And really strangely, and this is when people did genome sequencing, they found that there were a few organisms that actually had less than that, which is really bizarre. And so what's going on there? Turns out that there are a few organisms, particularly bacteria and Archaea, were the sort of really obvious outliers here, where you have an amino-acyl tRNA synthetase that puts one amino acid on a tRNA, and then a second enzyme that comes in and changes that amino acid. So you can actually have fewer amino-acyl tRNA synthetases. You have an amino-acyl tRNA synthetase. In this particular case, it's glutamine and glutamate. So one amino-acyl tRNA synthetase that puts on glutamine and glutamate, and then another enzyme comes in if it's a glutamate tRNA that will change it from glutamate to glutamine. So you end up with this changing that happens here. But because of the fact that you've only got on the order of 20 amino-acyl tRNA synthetases, that means they have to bind to multiple different tRNAs. This is a crystal structure of what one of these looks like. I don't think this particular one's from the Perona lab, but there are a number of crystal structures of these things from the Perona lab, where basically you can see the protein in gray, the tRNA in purple, and here is where you have the modification that takes place where you put that amino acid onto the tRNA. Main thing to emphasize here is there's lots of interactions between the tRNA and the protein, not just at this anticodon loop, but all throughout the tRNA. So the whole thing has got interactions here. What does this enzyme do? It takes an amino acid, and by hydrolyzing ATP, hooks it up to the OH present at the CCA 3' end of your tRNA. In that way, it activates the amino acid because you have this ester linkage now between your amino acid and your tRNA. There are actually two steps in how this happens. First, the amino-acyl tRNA synthetase adds this to an adenine and then transfers that to the tRNA, which is at the end. And so this is a two-part reaction, and it turns out there are different flavors of amino-acyl tRNA synthetases that do these in two slightly different ways, having to do with whether you add on to the 2' OH or to the 3' OH, which is at the end of your tRNA. It always ends up on the 3' OH, but in some cases it gets put onto the 2' OH to start with. And those are the two different sort of classes of amino-acyl tRNA synthetases. Because you've got an amino-acyl tRNA synthetase and you have the tRNA messenger RNA interaction, a lot of people talk about this as being sort of two different adapters, trying to figure out how you get this translation, again, from nucleic acid language to amino acid language taking place. Your amino-acyl tRNA synthetase recognizes a particular tRNA or classes of tRNA that need to have one particular amino acid that's associated with them. And then that tRNA, once it has the amino acid associated with it, now needs to go and bind to the messenger RNA and incorporate that messenger RNA, incorporate that amino acid at the appropriate place in your protein. And so they're really these, you've got the amino-acyl tRNA synthetase that says, okay, put this particular amino acid on this particular tRNA, and then that tRNA goes into the ribosome and has to find the appropriate codon-anticodon interaction in order to get that amino acid put in. So there are two separate control steps here to figure out what's going on, and a lot of that then leads to the appropriate specificity. And again, getting back to this question about whether the tRNAs could function with these extra pieces in them, they've got to be extremely similar in terms of their structure in order to have a pretty non-specific machinery, the ribosome, be able to add pretty much any sequence of amino acids that it could. So how does it make sure that we've got the right amino acids which are put on the end? This is an analogous process to what happens in replication when you have proofreading. Incorrect base pair, that gets cut off. Here, if you have the incorrect amino acid put on the end of your tRNA, that gets cut off. The way that that works is you have an editing site, and if you've got an incorrect amino acid, that then gets chopped off. It's the correct amino acid. It actually gets left on. It doesn't get cleaved off. Now, why is this such a big deal? Well, if you think about the amino acids, there are lots of them that are really, really similar to each other. My favorite example is actually tyrosine and phenylalanine. They use leucine and isoleucine and valine here. But these guys have practically identical structures with one little tiny change at the end there. And this is the original first synthesis reaction, which takes place on your amino acyl tRNA synthetase, adding on that particular amino acid. That can very often end up adding the wrong one. So you'll have a tyrosine amino acyl tRNA synthetase that puts on a phenylalanine because it looks pretty similar. But then this phenylalanine gets chopped off because in this particular case it fits into this editing site. Whereas this OH at the end there blocks that particular amino acid getting into the editing site and getting chopped off. Yeah?
Is that editing site in the amino acyl tRNA synthetase or in the tRNA?
So the editing site is in the amino acyl tRNA synthetase. So it's the part of the enzyme activity which is there. And so you can also think about this like, again, we'll use the polymerase, the DNA polymerase analogy. It has a polymerase active site and it has an editing site. And these are two separate sites, but they're still in the same molecule. So same thing here. You've got our synthesis site where you add on that amino acid, and you have an editing site which is in a different place, and that's why, sort of highlighted, which is right here. Because of this, you only have about a 1 in 1,000 error rate, and apparently a 1 in 1,000 error rate is good enough for protein synthesis. And we'll see that we have some issues with this a little bit later on. One of the ways that people were actually able to show that you have this two adapters is that you can purify tRNAs that have amino acids that are attached to them and then change that amino acid and then see what protein you actually end up with afterwards. And so this was the original experiment which was done where people took tRNAs that had cysteine attached to them, purified those tRNAs, and then did a chemical reduction where you get rid of this SH group at the end of the cysteine and end up with alanine, and then put these cystenyl tRNAs that have alanine attached to them into a normal translation reaction like the one that I talked about Nirenberg did with poly-U. And then you look at what amino acids get added. And it turns out that the ribosome could care less what amino acid is hooked up to the end of the tRNA. So in this case, if you had cysteine codons, you still end up with alanine in this particular case. So what is this reaction that takes place in the ribosome? Also known as transpeptidation. Basically, you have your amino acyl tRNA, which then brings in this activated amino acid next to a growing chain, which is started at the N-terminus, moving to the C-terminus. So this is the molecular biology polarity, going from N to C. Adding on at the C-terminus, this now adds to a peptide chain which is bound to the 3' OH of a tRNA which is already there. And so what you're doing is you're just exchanging this peptide chain, or the individual amino acid which is there, you're moving it from one tRNA, in this case tRNA 3, to tRNA 4. And so these get attached. Your polypeptide chain is being extended here while it's on one of these tRNAs. So your peptide chain is being elongated from N-terminus to C-terminus by addition at the C-terminus by a tRNA bound to one of these amino acids. But if you remember, where's that high energy bond? Where's that high energy bond? Where's that formed? So the high energy bond is at the 3' OH. So where is the energy coming from for this reaction? It's coming from the long peptide. It's the one which you've already started with. So this is now the opposite of what's happening when you add nucleotides with DNA polymerases or with RNA polymerases. So DNA polymerase and RNA polymerases, you're getting the energy from hydrolysis of your triphosphate. The triphosphate's what comes in, that gets hydrolyzed, now you get the elongation of your chain. Here, on the other hand, the high energy bond is present in the long piece that then gets transferred to your short piece, which is now the long piece, and extension. So people talk about this as head polymerization as opposed to tail polymerization. Again, those terminology is not important, but the important thing here is that you have the high energy bond, which is in the long piece, as opposed to the high energy bond, which is coming in the individual monomers. So that's the transpeptidation process. Again, as a reminder, we're talking about peptide bonds here. I'll also talk about these as the backbones of your amino acids. Each of these has a different side chain, R1, R2, but you're always going here. It's called the alpha carbon, beta carbon, which is the carbonyl attached to the amino group, etc. And always have a N-terminus and a C-terminus. You're always adding to the C-terminus. How does this work? We have our messenger RNA, which you have to get the ribosome associated with. That ribosome needs to associate with the initiator tRNA, which 99 times out of 100 is going to be methionine. Then you get the large ribosomal subunit, which basically clamps down on the messenger RNA with this tRNA in it in the first place. And then you have extension that takes place. The RNA probably moves through the ribosome. Lots of people talk about the ribosome moving on the RNA. It's much more likely the RNA is moving through the ribosome, very similar to what's happening in transcription, replication, etc. And you have this growing polypeptide chain until you get to a stop codon. Everything dissociates, and you go around again. Again, this should look extremely similar to what we see in terms of transcription. Initiation, elongation, termination. So let's look at the individual pieces that you have here. We talked a little bit about the different RNAs that you need to make your ribosome with. We talked about RNA processing, the ribosomal RNA processing. You have these RNAs, again, that we talked about last time. 28S RNA, 18S RNA, 5.8, and 5. These are just differences in terms of their relative sedimentation coefficients, which depends on their size. And so a lot of people in office hours ask me, what the heck is this S all about? Well, the S is all about how rapidly these complexes sediment in an ultracentrifuge. And so that's where those S numbers come from. It's just how they were seen. This particular band right here was at 70 Svedbergs, which is what the S stands for. This one's 50. This one's 30. If you add up all of these together, they still don't linearly add up together with each other. And so we'll look at the prokaryotic ribosome here. This would really be bacterial ribosome. You have a small subunit at 30S and a large subunit at 50S. You add them together, you get 70. Okay, wait a minute. 30 plus 50 makes 70. Okay, but that's just because it's a nonlinear relationship between these two, and the reason they're called this is how people actually purified these things. They put them in an ultracentrifuge and spun them and pulled out the band that was at a certain relative migration. And so you had here 30, 50, and 70. And that's what you end up with. And so the numbers are not going to add up here. Important for us is not so much exactly what those numbers are. I can't remember what they are all the time, so I can't expect you to remember what they are. But there are a couple things that I would remember. One is that it's all about the RNA. And so if I'm talking about the RNA, 16S here for the small subunit RNA sequence in bacterial ribosomes, 18S that you have in eukaryotic ribosomes. People, I don't know if any taking microbial ecology or microbiology right now? Yeah, a couple of you. So you probably heard about 16S and 18S, probably a whole bunch. That's because these are also great molecular markers. And you remember some of those trees we looked at way at the beginning of the class in terms of archaea, bacteria, and eukarya? Most of those original trees were based on these sequences, on the 16S and the 18S sequences, in terms of how different they are from each other. The large subunit is either 28S or 23S. And so those are going to be the ones that I would expect you to know. So these then come together, make these larger complexes, 70 versus 80. Again, the numbers there don't matter. But the important thing is the large subunit is where you have the business end of your ribosome. That's where the peptidyltransferase activity takes place. And the small subunit is where you have codon-anticodon interactions. And so that's, again, the important thing which is there. Now let's actually look at the structure of the ribosome. Most people say this is a prokaryotic ribosome structure. So the first ever ribosome structure of any of the subunits was, in fact, from Archaea. Woohoo, go Archaea. Particular ones from an extreme halophile. But the take-home message here is that everything that's in gray is RNA. And hopefully you can see it's mostly helical. All these helices all wrapped around each other. And then the proteins are sort of decorations hanging out on the outside. Probably important for stability, but clearly a very long way away from the business end of the ribosome. And right here in green, it may be a little hard to see. Here, I guess you can see it reasonably well. That's where the transpeptidation reaction takes place. And here, if you look where that particular place is, there are no proteins within miles. Well, okay, it's angstroms. But they're a really long way away from where the actual active site is. And people had done some experiments where, even before this structure, they had tried to get rid of as many of the ribosomal proteins as they could and still see if they had activity. And so they actually had a really good indication that you could get rid of almost all the proteins and still have ribosome activity, still have this activity of linking up the amino acids in order to get a chain together. So that's the original large subunit structure. This is now looking at this in a color-coded form. But again, the main thing here is it's all about the RNA. This particular one is, let's see, I think this one's from a bacteria rather than an archaea here. But all of this, it's all RNA. You can look at the structures, break them down with this base pairing interactions, the helices and loops. That's just shown over here on the right-hand side, each of the individual domains. And because it's all about the RNA, a lot of people talk about the ribosome as being a molecular fossil. Why? Because people think that life, as we know it, really started with RNA. So it was RNA which had all of the enzymatic activity. It was RNA which had all the genetic information in it. And then going from RNA to protein, which is what the ribosome does. That's what it is. It's a amino acid polymerase you can think of as well as what you want to call it is a ribosome. Basically, it's RNA which you need to make protein. So RNA, in this case, clearly came before protein. So again, that's why this is sort of the molecular fossil. Turns out that proteins do a lot of enzymatic activity a lot better than RNA does. And so that's evolutionarily probably why most enzymes now are made up of protein rather than nucleotides. It's just another view of looking at this. Ribosome, here's the large subunit of the ribosome. Here's the small subunit of the ribosome. These three are individual tRNAs. Once you flop this small subunit of the ribosome on top of the large subunit of the ribosome, you basically can't see these guys anymore. And that's because they're really trapped right in the middle of the ribosome. There are 3 tRNAs that are present in the ribosome. There are 3 sites for tRNA binding. They are the A site, the P site, and the E site. A is the aminoacyl site. That's where you have the tRNA that comes in with the single amino acids attached to it. The peptidyl transfer site, which is the one here in the middle, and the exit site. And so what that looks like, we'll take a look at the animation here. Oops, back up. Let's not click on that. And that will be how we finish. Oops, come on. Escape from here. Go back one. This is the animation of translation, which you can also apparently link to on your textbook site. I've been told that you can do that. I tried to add it into the PowerPoint presentation, and it didn't like me. So we'll try this one instead. If it will actually load. Here we go. The job of this mRNA is to carry. So here we have the messenger RNA, which was made from our RNA polymerase. Remember, it's zipping along its DNA. So now it's exported out into the cytoplasm. And here's our ribosome chunking along this messenger RNA, producing this protein right here in red. And again, so what we should be obvious here is this is coming out of the structure where you've got the individual pieces which are present there. So there's the small subunit of the ribosome, binds actually to the end. The large subunit comes down on top of it. And now we're zooming in at each of those individual nucleotides, 3 of them together. Here are all our tRNAs floating around with their little red noses. I guess it's Rudolph time. That's supposed to represent the amino acid. And again, that's what ends up in that protein which gets produced at the end. So each of these aminoacyl tRNAs comes into the A site in the ribosome, transfers to the P site. That's where you have transpeptidation that takes place, and you make the extending amino acid side chain as you're moving through here. So that's that blow up here. We're just looking at the large subunit here. The small subunit will be sitting down on top of this while these interactions are taking place. And again, it's all attaching from the new tRNA that comes in as its amino acylation. That gets hooked up to the C-terminus of the polypeptide chain, which is already in the P site, and that's how you're getting extension taking place. So with that, I think it is now 10:05. The rest of this is actually looking at the extension of the amino acid side chain, which ends up folding up and making a protein.